Thank you very much, Zev. And it is such a pleasure to be here. I want to thank Milton and Zev and everyone on the organizing committee for inviting me to speak today. And I should apologize in advance because I'll be a little bit provocative, or maybe more than a little bit provocative, because the title here is Testing the Boundaries. I wanted to ask, to what extent might we be violating the boundaries? And are we doing things that in retrospect may become a problem for us? Let's see now, how do I get started here? Eek. Before I begin, I want to acknowledge MC Chang. And for the young people in the audience, you may not know the name MC Chang, but we all stand on the shoulders of great, great scientists. MC studied in Tsinghua University in Beijing. He ultimately came to the United States and had a career at the Worcester Foundation. It was his work that led to the discovery of the birth control pill. And if you think about life today, it's hard to imagine it without women being able to control their reproductive cycles. He also discovered capacitation in sperm. And with one of his students, Yanagamachi, they developed in vitro fertilization in the mice. And MC was never really fully acknowledged the way he deserved to have been, but it's absolutely remarkable, the work that this great Chinese scientist did in a cooperation involving Chinese-American partnerships. And it's one of the real joys that brings me here, because between Asia and the New World and the Old World, we have so many friendships and cooperations, notwithstanding what my president says. I need to tell you that I've gone to China long before many of you were ever born. So I first went to China maybe about 35 years ago. Back then, everybody was on bicycles. There were very few cars. And I told a joke, and I had a translator, and I loved it because I told my joke, translator translated it, and everybody laughed. And then that night, one of my former students came to the hotel. He said, "Jerry, you know what happened?" He said, "The foreigner told a joke, everybody laugh." And so everybody laughed. We are entering a very unusual world. It's a wonderful world, but it's a complicated world. And, oh, let's see, I guess I do it this way. And this is an image from New York Times of a person who asked their gynecologist to help them have a baby as they were going through gender reassignment. And so you can see this person who's going to become a man had stopped his testosterone. He had his biological son. He wanted to nurse him. And in the transgender community, they prefer not to say breastfeeding. They call it chest feeding. And then later, he will go on and have his elective surgery. It is a different world than the world many of us grew up with, and yet it is the world that we must embrace and look forward to. Maybe it's because of social media that people who couldn't find partners to love in the past are now able to find one another. But regardless, especially for many of you who are physicians, this is the world that you will be encountering. And this person, until he gets his surgery, you have to give him a pap smear. And in the same way, for women that transition, you need to remember to give them prostate exams. This is counterintuitive. This is different than the world that most of us grew up with, and yet it is happening in many, many countries. There are many other breakthroughs that are occurring that are transforming our field. For example, this is work that was done both at Rockefeller and in the UK, where embryonic stem cells and trophoblast stem cells were put together and an embryo was assembled. This embryo was never conceived. It was not fertilized. It was manufactured with different stem cell capabilities. The embryo will go through gastrulation, and the only reason it stops at 14 days is because of a convention to stop human development at around 14 days because of bioethical constraints. God knows how far these embryos would go in vitro. They may develop very far. And so here we have the possibility of manufacturing viable embryos, not through conception, but through mixing of stem cells. And at the other end of gestation, incredible things are happening. So for example, here, let's see, how do I do it? Can you see that? Yeah. There is a fetal lamb, but rather than delivering it in air, these researchers at Penn put it into like a Ziploc bag, and they use the fetal heart to pump fluids around through an artificial lung system. And this is the same lamb a month later. The lamb has wool, the lamb has meat on it, and, I'm sorry? Change the slide? No, it's over here. Can you see it? It's got wool, it's got meat, and the only reason they stopped the experiment was because, again, their animal use committee said they could grow it for 4 weeks in utero. Now, the day may come when a lot of premature babies that may be at risk for cerebral palsy or other disorders because of prematurity, may not be delivered into this toxic aqueous environment, but would be delivered into, toxic atmospheric oxygen, but into an aqueous environment where their lungs could mature, their surfactants could develop, and it really might transform what we all do. And yet, if the embryos go through post-implantation development later and later, and we can move placental development or in utero development earlier and earlier, will the day come when we can actually live in a kind of a brave new world of avoiding gestation totally. I also want to speak to the fact that at the end of life, we are facing a global crisis. All of us are aging, and our populations are aging. The young people in the audience are going to be paying enormous amounts of taxes to keep the old people out of nursing homes. And this is a huge problem in all of our countries, but it's the worst in China. Because with the one-child policy, many adult children have 2 parents to support and 4 grandparents to support. It ironically became a demographic upside-down pyramid. And unfortunately, you will need to grapple with that. And there have been wonderful insights into Alzheimer's disease. Alzheimer's is really what keeps us from enjoying life to its fullest. And I think you all know that a little over a century ago, a man brought his wife in to see Dr. Alzheimer's, and he said, "Oh, I can't put up with my wife anymore. She doesn't remember anything. She is so suspicious. She's convinced I'm running around with a neighbor lady. She's just impossible. What can you do?" And he took her into his clinic, and then eventually she passed away from dementia. And then he did the histopathology on her brain, and he discovered the amyloid plaques and the neurofibrillary tangles. What they don't tell you is after she died, he ran off with a neighbor lady. So she was right. I mean, that, they say even paranoids have enemies, but, she actually knew what was going on. And in the United States and in many countries now, there are enormous amounts of money to try to understand and to try to slow down the rate of Alzheimer's. Alzheimer's, is caused by neurofibrillary tangles, these byproducts of microtubules, and also amyloid plaques. And now it appears as if there are liquid organelles that can assemble within the cell, and normally they disassemble and then reassemble in different cell cycles, but not so in the aged brain. And if there are ways of slowing this down, we can solve the Alzheimer's problem by just slowing the rate of amyloid plaque and neurofibrillary tangle buildup. And I think there's great excitement about being able to slow the rate of aging. I do want to speak a little bit about some of the ethical tragedies that have occurred in our field. And many years ago at the Jewish Chronic Disease Hospital, some researchers who were interested in immunotherapy decided they would find dementia patients and patients with chronic debilitating diseases and inject them with live cancer cells. They figured, what the hell, they're going to die anyhow. But of course, that's true for all of us. We're going to die anyhow. And they didn't want to tell the patients that they were injecting them with live cancer cells because they thought, oh, they'll just get upset. And maybe rightly so. And I mean, ironically, it was one of the steps towards understanding immunotherapies, but it really is what led to informed consent. And so I think it's important for us to focus on what's gone wrong as well as what's gone right. There are amazing things happening in the aging field. And one experiment is done with an old mouse and a young mouse, and you mix their bloodstreams. It's called heterochronic parabiosis, and material in the blood system of the young mouse goes into the old mouse, and it rejuvenates the old mouse. So there are factors that are circulating in the blood system of young mice that rejuvenate old mice. And Wall Street is all over this. There's a company in San Francisco now called Embryos. For $8,000 a week, I can get the plasma from undergraduates, and they claim that it will restore my vitality. And you know, there's historical evidence for this because Dracula never sucked the blood from old people. Dracula always went after young people, right? I mean, so, but there are factors that are in the blood of younger people and animals that seem to slow down aging. Now, I want to talk about aging from an embryological perspective. And let me start by saying, and I may have to violate the microphone here, but you know, we all start as gametes. The gametes come together and they go and become an embryo. You get implantation, you birth, eventually you go to medical school and you get a grant or two, and then you die. And it's a one-way highway, a one-way trip from gametogenesis to death. It never is reversed, except every now and then. So remember with Dolly, right? There was took a lot of experiments, and Dolly started as a mammary gland cell from a 7-year-old ewe, and that cell was put into an unfertilized egg, and in an almost miraculous way, time reversed, right? Instead of having a 7-year-old sheep, you started the clock over again. It's like in Monopoly, when you go around Go and you collect $200, it's just like that. Somehow that happened. And it was really quite miraculous at the time. And that led to the whole field of induced pluripotent stem cells. And we're going to be hearing more about the plasticity of these cells in the afternoon. But it's really quite remarkable that you can take cells from the skin of people in their 80s and turn them back into embryonic cells. And if you can do it in vitro, well, maybe someday we'll be able to do it in vivo. Maybe someday, instead of transplanting kidneys or livers, we'll be able to induce the cells that are damaged to repopulate the scaffold of the organs that are damaged. So the whole field of regenerative medicine is really quite exciting, and it's moving along very quickly. There are some wonderful things that are happening in regenerative medicine. But if you have a loved one who is ill, do not believe what you hear on the internet. There is so much crap about clinics that are offering all sorts of magical things that are not are not really parts of medicine. They're hype. For example, here, this is one that talks about your monthly miracle. Guozhang, this won't work for you. Don't try it. Right? I mean, and, you know, the women in the audience will appreciate that, okay, this is targeted to them. But even so, there are probably better sources of stem cells. Aging also affects us in terms of our field of reproduction. And there was a brilliant Harvard professor who discovered, he said, the reason for female infertility. The culprit, he said, was the education of women. It diverted energy from the reproductive machinery to the brain, resulting in women with monstrous brains and puny bodies. Now, let me remind you, no man has ever diverted energy to his brain, and certainly not this guy, right? But the issue of reproductive aging in women is really quite amazing. And from what we heard from Norbert and others is that it really boils down to the age of the egg. There are some people following on experiments that Jacques Cohen originally did who are convinced that eggs have mitochondria that get tired. So I've heard from Bob Casper and others who talk about the importance of supplementing mitochondria in the egg. And what they tell you is they say, look, you know, if you leave a flashlight in the closet for 40 years, the batteries run dry, right? All we're doing is changing the batteries. And it sounds really good. And people have signed up for the clinical trials in Canada and I guess also in the Emirates and a few other countries. And their notion is that, well, mitochondria are just like batteries. And if you can replace the batteries, great. However, it's much more complicated than that. I'll come back to this in a second. There are a lot of mitochondria in a cell. Don't think that there are just a few mitochondria. In some eggs, 99% of the DNA is mitochondrial DNA. So mitochondria play an important role in roles that we haven't fully understood yet. And the United Kingdom, the United States, and many countries has had a very spirited, thoughtful discussion about how to help women who have mitochondrial DNA mutations. Now, this is a small percentage of the population, and there have been really an awful lot of deliberations, and the idea is that it might be possible to get an egg from a healthy donor. You will end up with a fertilized egg in which the nucleus, the spindle, comes from the woman who wants to be the biological mother. The father's sperm comes in here, and you end up with the cytoplasm from the donor. So this is often referred to as three-parent IVF. I think for Hallmark cards, it's going to be great, because on Mother's Day, you can have a card from my nuclear mom, and then another one from my mitochondrial mom. They can double their sales. It's great. Okay, thank you, Milton, for laughing. But this has been a topic that has had a very spirited, lively, informed debate about whether it's appropriate or not. And I'm sorry John Chang isn't here, because John is the pioneer who's given, he has not personally given birth, but he has helped the couple give birth to a boy derived from oocyte spindle transfer to prevent mitochondrial disease. Now, I want to commend them in so many ways. One way is that they worked with a couple that had this unusual defect that many groups agree is worthy of this special therapy. This couple had had many miscarriages. They also lost at least one or maybe two children in early childhood. Furthermore, they only transferred male embryos. And because of the maternal inheritance of mitochondria, the idea is that you're preventing inadvertent germline gene therapy. If it's a boy, you would not expect to have the mitochondrial DNA transfer for more than one generation. And so I think this is commendable. The way the paper was written, it appeared as if they did the embryo work in Mexico because it's not permissible in the United States. Norbert published something that he questions whether they actually did it in New York. And I think New York is still part of the United States. It's not like Puerto Rico, right? It's still part of the US. No, never mind. Now, since this work has come out, there have been other groups around the world that have made girls as well as boys through this technique. And I worry about the effects on those daughters. And also there are groups that are using this not for patients who have mitochondrial DNA mutations, but for just general IVF patients. I've heard groups talk about, well, maybe this would be a great therapy for lesbian couples because in principle, in principle, you could have one member of the partnership donate the cytoplasm and the other member of the partnership donate the nucleus and then get donor sperm. To me, this is crazy. There is no reason to be doing spindle transfers and mitochondrial transfers when you have perfectly healthy eggs. I mean, doing these kind of medical experiments on helpless couples seems really unethical. We know a lot about mitochondria, but there's an awful lot that we don't know. I like this experiment actually that came out of China, in which a mitochondrial protein that's involved in generating ATP is overexpressed in one set of pigs, this pig here. And you can see the pig is hotter than the other pig. This pig is generating a lot more heat because the mitochondria are uncoupled. And as a result of that, there is less fat and more meat. It's using the technique CRISPR, and they call it crispier bacon. In principle, you could also use this with us. You know how babies are born with brown fat that generates heat? You could introduce, in a way, what's called beige fat, where our fat could have a higher mitochondrial concentration, and we would become less obese. And obesity is a global problem. It's amazing. Now, this had been used back in the 30s. There was a treatment for weight loss. It involved dinitrophenol, and it broke down the proton gradient that made the ATP in the mitochondria. And it was fabulous because you lost so much weight. Now, there was one problem. It was irreversible. So you died, but you looked great at the funeral. I mean, it just, boy, really hot. Well, ironically, really hot, but there you can see the heat. Mitochondria are incredibly motile. So these are mitochondria in chlamydia. Mitochondria also traffic to the microtubules of the mitotic spindle. So as the cell divides, you want to see that again? Yes. Okay. As the cell divides, the mitochondria are on the microtubules. So the microtubules form, of course the chromosomes align, and then the cell divides. But what you have to appreciate is it's not just the chromosomes the microtubules are segregating, but it's all the contents of the cytoplasm, and in fact, different mitochondria bind to microtubules in different directions. So in some cases, one set of mitochondria ends up in the daughter cell, and a different set of mitochondria ends up in the mother cell. So just because you start with uniform cells, don't think you end up with the same pattern of mitochondria. There have been lots of studies done on mice and other systems, and mitochondria behave in ways that we really don't fully understand. And so be very cautious when you hear somebody say, oh, they're just batteries, and we're just going to replace the batteries. It's much more complicated than that. Now, I want to switch over to designer babies, and this is an enormous area, growing by every day, and, whoops, there's no slide back here, it's over there. And, is there something, do they translate things into Mandarin? No, okay, thank God. So there's a clinic here that's offering to help you select the IQ of your baby. You could have an IQ of a 70, or your baby could have an IQ up to 130. And if it's 130, your baby might become an attorney or a chemist or an executive. Milton, no doctors. Rob, no scientists. I'm sorry. But they'll tell you that, you know, your baby might live in poverty and have 32, I guess that's 32% will have illegitimate children, or is it 32 illegitimate children? It goes on and on and on with all these details. Divorced in 5 years, yeah, talk to some people. Anyway, unemployment. I mean, this is crazy. And yet people are offering to raise the IQ of your offspring. This does not exist. Fake news. But yet it is being marketed for an extra $25,000. And what they're doing is they're taking the DNA from mathematicians who are 3 standard deviations above the norm and claiming that if they can help select for that, your baby will be of a higher IQ. My wife is a mathematician who is 3 standard deviations above the norm. Not everybody is going to want to have the father or mother of their children being a mathematician 3 deviations above the normal. Most mathematicians are really weird. Most mathematicians, when they give a lecture, they give a lecture to their toes and they don't talk like this. If you find an extroverted mathematician, they speak to your toes, not their own toes. I mean, you don't necessarily want a mathematician to be the parent of your offspring, and yet this is being marketed now. And in an article in the New York Times, now there's a certain number of genes that are associated with human intelligence, and the technologies are coming along so fast that this is not something that we can necessarily laugh about, because it is happening now and it's happening very swiftly. So there are ways in which various animals can be treated to double the muscle mass. I think this is using myostatin, same breed of dogs. And it involves the CRISPR technology. Now, CRISPR is amazing. And CRISPR is a technique that doesn't necessarily change the DNA, it erases the DNA. And because it's an erasure, it doesn't count as genetic modification. So, Mark, I brought an apple. It's a ripe apple. And there's a company now that will be marketing Arctic apples. And these are apples that have been used, that have had the CRISPR technology used so they won't brown. You can bruise them and they won't brown because they've knocked out the browning enzymes in the mitochondria. And you don't have to disclose that to the USDA or in Europe. There are mushrooms that don't brown. There are all sorts of different organisms that are being made without, you want the apple? You could use it for your next talk. I'll leave it there, okay. And it involves this amazing technique that is called CRISPR, and I hope we have the sound up high.
The CRISPR method is based on a natural system used by bacteria to protect themselves from infection by viruses. When the bacterium detects the presence of virus DNA, it produces 2 types of short RNA, one of which contains a sequence that matches that of the invading virus. These 2 RNAs form a complex with a protein called Cas9.
Now that's a guide RNA and Cas9.
A type of enzyme that can cut DNA. When the matching sequence, known as a guide RNA, finds its target within the viral genome, the Cas9 cuts the target DNA, disabling the virus.
So this is a double-strand break in the DNA.
Over the past few years, researchers studying this system realized that it could be engineered to cut not just viral DNA, but any DNA sequence at a precisely chosen location by changing the guide RNA to match the target. And this can be done not just in a test tube, but also within the nucleus of a living cell. Once inside the nucleus, the resulting complex will lock onto a short sequence known as the PAM. The Cas9 will unzip the DNA and match it to its target RNA. If the match is complete, the Cas9 will use 2 tiny molecular scissors to cut the DNA. When this happens, the cell tries to repair the cut. But the repair process is error-prone, leading to mutations that can disable the gene, allowing researchers to understand its function. These mutations are random, but sometimes researchers need to be more precise, for example, by replacing a mutant gene with a healthy copy. This can be done by adding another piece of DNA that carries the desired sequence. Once the CRISPR system has made a cut, this DNA template can pair up with the cut ends, recombining and replacing the original sequence with the new version.
So as we get towards the end of 2017, you'll see that many of the breakthroughs in 2017 will be the result of the application of Cas9 and CRISPR technologies in human embryos. So there's been some work that was done by Shrukat-Militipov's group, and there have been questions about some aspects of it, and yet by and large it's going to hold together questions about whether or not the paternal DNA played as active a role as the maternal. There's also been groups in the UK that have used OCT4 to be knocked out in human embryos using CRISPR, and that work has been substantiated. And then there are groups here in China who have worked with patients who have thalassemia, and they grew the thalassemia patient cells in a dish, then used CRISPR to correct the defect in the tissue culture cells, and then used cloning techniques to clone the corrected nucleus back into the human embryo. And so work in this field involving genetic manipulations and also nuclear transfer cloning is moving at light speed in human embryos. And you'll see that the bioethicists will be asking powerful questions. Procreative beneficence, why we should select the best children. There'll be others that this one just came out. Do we have a moral obligation to genetically enhance our children? So this is turning it around. It's not a question, should we not, but should we? And then there's also a question about a right not to be born. I won't dwell on this. But let me just say that we all see the world through very different lenses. We all are raised in different faiths, in different cultures, and yet as doctors and scientists we can communicate with one another. We can disagree on what we disagree on. This image here, when you show it to children, the only thing the children see are dolphins. It's only people with filthy, filthy minds. Nava, do you see any dolphins? No, no dolphins. Okay, let me show you. There's one dolphin down here. Oh, you people have filthy minds. But I mean, I like this image because it says that we see the world in a way that we're programmed to see it. And oh my God, I don't want to say too much about this audience. But we see the world depending on our faiths, traditions, and our background. And there was an article recently in Nature about how China, using PGD, is pushing to avoid a whole set of genetic diseases. I don't know to what extent this article is true. I don't know to what extent we all share the same views about whether children with genetic diseases should be born or not born. You know, one hears about people in the deaf community who are seeking embryos that they know will be deaf because they have a rich tradition. So there's many complicated things in this area, but one thing that's happening, and it's a paper that came out yesterday in the journal Cell that is totally transformative, involves epigenetics. Now let me remind you that epigenetics involves a number of different modifications on the chromatin. And the chromatin involves both the DNA that comes from both parents, as well as the histones that are the core structure onto which the DNA is wrapped. Now, there are a variety of modifications in DNA and modifications in histones, and a brilliant paper came out yesterday involving the use of Cas9, but what they did is they used a dead Cas9. So this Cas9 cannot make that double-stranded break in the DNA. So rather than destroy or modify the DNA, they used a dead Cas9, and rather than using a guide RNA for a particular sequence, they used the guide RNAs that would only find imprinted regions. They incorporated this dead Cas9 CRISPR technology into mice that had a variety of diseases. In one case, it was a muscular dystrophy mouse. In another case, acute kidney disorders. In another case, diabetes. Now, let me remind you, this CRISPR technology is not touching the DNA. It is only influencing the epigenetic imprints. It is brilliant. It is an epigenetic modification. And when they do this, this is one example of this mouse has muscular dystrophy. This is the same mouse in which they injected the hind limbs with the epigenetic modifiers that will increase muscle cells. So in the same way, you can see that this is the control, and this is the muscular dystrophy mouse that's now been induced to grow more muscle fibers. And you can see the muscle fibers are so much bigger. So this is likely to open up a whole new field of medicine, epigenetic pharmacology. There are histone deacetylases. There are histone methyltransferases. There are DNA methyltransferases. There are all sorts of drugs that are moving through the pipeline that will be modifying the epigenetics, not modifying the chromatin as the original CRISPR thing. I'm not going to spend much time about germ cells because we have great lectures later today from the pioneers who were able to take embryonic stem cells or iPS cells and make sperm or eggs out of them. And here you have a slide where Noah is talking to all the animals that are going to want to go on the ark, but Noah says, nope, I'm only taking tissue samples. And almost every zoo now is taking skin samples because they hope to make gametes from endangered species. But here in the audience, we have a lot of young people, and I want to speak to the young people about how you are endangered species. And let me tell you that about 10 years ago, the Nobel Prize was given to 3 people. Only 3 people, living people, can get the Nobel Prize. It was given for green fluorescent protein, for GFP. And they noted that the man who actually had done the sequencing of the green fluorescent protein was this guy, Doug Fraser. He could have gotten the Nobel Prize, and they could have easily let us out, Marty Chaffee said. When the Nobel Prize was awarded, he had lost all of his grants. He couldn't find a job. He had spent his life savings, and he was driving a shuttle bus for a Toyota dealership in Huntsville, Alabama. He was this close to a Nobel Prize, and he was driving a shuttle bus. And so for the people from the magical Sun Yat-sen University, please make sure that you have a career after you're done with your job, with your studies. Many people study in such a narrow area and never end up with the job of their dream. So make sure you have what we call a plan B. And for the physicians, you always have a medical license. But don't be lost by spending all your time in the lab and not thinking about your livelihood. I should also mention that Roger Chen, who did this work, passed away a year back in a bicycle accident. Now, I want to end by saying, years ago, I thought I could see clearly the ethical issues. And this is a friend of mine, Tom Starzl, who just passed away. Tom did many of the pioneering studies on liver transplants. He killed a lot of people. He had a lot of patients that did not survive. And probably in today's climate, he would never have been able to do the pioneering organ transplantation that so many people benefit from nowadays. At first, when he was doing this, every religion went after Starzl and said, you are desecrating the dead. You are ripping the warm organs out of my loved one. I mean, this is an abomination. This is not medicine. And then as people began to survive, people began to realize that organ transplantation really does have a place in medicine, but very few people were at the front of that, that parade. And about 10 years ago, a few of us spoke at the United Nations to ask them not to block somatic cell nuclear transfer. At the time, Christopher Reeve, who was then known as Superman, was going to speak with us, but his health was declining. And so I'm going to end by allowing you to hear what Christopher Reeve said just before he passed away.
In person. Now, as representatives to the UN, all of you hold positions of enormous responsibility. You are perceived as the voice not only of the countries you represent, but as the collective moral voice of the world. Millions of patients just like me will be watching with great concern as you continue your important deliberations. Everyone I know of, including the scientists you'll hear from today, opposes the cloning of babies and the pursuit of reproductive cloning. But every leading scientist seeking cures calls for stem cell research to advance. Therapeutic cloning, also known as somatic cell nuclear transfer, offers real hope. The research is still in its infancy, but it has so much promise. And not to encourage the ethical pursuit of this research might result in needless human suffering and destroy the hope of those afflicted with Parkinson's, Alzheimer's, spinal cord injuries, and a host of other conditions for which no cure is yet known. Countries around the world are grappling with this issue and deciding that the purpose of government is to do the greatest good for the greatest number of people. Countries can ban reproductive cloning and still live up to their obligation to provide the best medical technology to all citizens. And I have a real concern that a great medical advance might be lost to humanity should the United Nations recommend a treaty that would prohibit this research. So my prayers are with you, and I hope that you'll make the right decision. A decision based on secular law and morally sound scientific knowledge that will provide hope to millions suffering all over the world. Thank you very much.
Thank you very much. I appreciate your attendance.